Hello everyone, welcome back to my YouTube channel. So after the confirmation about the genetic material, that DNA is the genetic material, the question came in the mind of the people that how the DNA will be fitted inside the nucleus, because the size of the DNA is so long. The DNA thread is so long. In case of eukaryotic organisms, it is almost 2 meter. It is 2 meter in case of eukaryotic organism. And it was known to the scientists that the DNA is situated inside the nucleus. And the size of the nucleus is almost 10 micrometer. So can you see what is the size difference? Inside such a tiny cell organelle, how 2 meter long thread will be fitted? That was the utmost important question which came in the mind of the scientist. So the only concept which came in their mind, that was coiling or condensation of the thread-like structure, and by coiling the condensation is possible, and once they will be condensed so much, then only that 2 meter long thread can get fitted inside the 10 micrometer sized nucleus inside such a tiny cell organelle. So from there, the idea of nucleosome came in the mind of the scientists. Before discovery of the nucleosome, or before saying this nucleosome model, or before coming into this concept, firstly the DNA and the protein which was present in the nucleosome model, that were known as new body, NU, new body. So firstly it was known as new body. After that, one scientist, that is Outdate, and so many other scientists gave the term, for the first time they gave the term nucleosome. And after that, Roger Kornberg was the scientist who actually gave mostly accepted model about the arrangement of the chromosome inside the nucleus, that is nucleosome model. So what is that nucleosome model? Let us now begin with that. So firstly, let me just draw one picture. That is, when they saw, they told about the nucleosome model, what are the components of the nucleosome model. So basically the DNA is made up of, we have already seen the structure of the DNA or chemical makeup of DNA, that is polynucleotide. It is consisting of polynucleotides. And one nucleotide molecule is consisting of sugar, base and phosphate. Among all of these molecules, this phosphate is nothing but the phosphoric acid. And due to having this phosphoric acid, the DNA is extremely acidic in nature. So if these are the threads of the DNA, these are the total threads of the DNA. So coiling is only possible if something is there, because coiling means it needs to be like this. It needs to be like this. Now if two negatively charged molecules will come close to each other, they will never come close to each other, because same charged particles never attract each other. They will repulse back. So in this way coiling is never possible. So the concept came in the mind about the histone protein, about the protein which is a basic kind of protein. So there was a protein which is known as histone protein. So as it is a protein, the building block of protein is amino acid, and as I said that histone is a basic kind of protein, so there must be some alkaline kind of amino acid which will be present over there. So mainly there were two types of amino acids which were present. One is lysine and the other one is arginine. One is the lysine and another one is the arginine. These two basic or alkaline amino acids are present in the histone protein, and according to the different molecular weight and according to the presence or amount of lysine and arginine, the histone also has several subunits. 5 types of histone proteins. The presence of 5 types of histone proteins were being marked. H1 histone, H2A, H2B, H3 and H4. These were the 5 types of histone proteins, the presence of which were marked by the scientists. So these are the types of histone proteins depending upon different types of their molecular weight as well as the amount of lysine and arginine in them. Now, how all of these are arranged? These are arranged in somehow like beads on a string kind of a structure. Beads on a string kind of a structure, like this is the string, this is the bead. This is the string, this is the bead, this is the string, this is the bead, this is the string, and in this way this will be continued so on. So these beads are nothing but these histone proteins. Beads are these histone proteins. So how are they arranged? Now let us see that. Say for example, this is one H2A. So it has actually another copy of it. That means it is forming dimer. So let me just draw right behind it. There is another copy of H2A. After that, say for example, this is H2B. That means it is also forming a dimer. So that means here once again another histone, H2B is present. H3 protein. If this is the H3 protein, it is also forming a dimer. So exactly behind it there will be the H3 protein, and if this is the H4, then it is also forming a dimer. So right behind it there will be the H4. So that means in this way, how many molecules of histone proteins are present there? 1, 2, 3, 4, 5, 6, 7, 8. So in this way they were forming histone octamer. Histone octamer. I have not talked about nucleosome till now. This is forming histone octamer in this way. So each will form dimer. That means H2A, H2B, H3 and H4. All of these will form the dimer, and in this way accordingly they will form tetramer, and 2 tetramer will be attached together to give rise to the octamer, and they are exactly present like this. Exactly they are present like this. From the front portion if you will see, then you will think that there is only one molecule. But when you will see from the side view, then you will get to understand that there are 2 molecules. These are the dimers. So in this way, there were 8 molecules altogether which is forming histone octamer. Now, this is exactly basic in nature. So it was helping the DNA, that is the thread kind of a structure. So it helps the DNA to wrap around it. So DNA will wrap around it in this way. DNA will just wrap around it in this way. In this way. This blue colored portion is the DNA. So this much is the total. This is the total round, and after that the round is finished. So in this way the DNA will wrap around the histone octamer. This is the DNA which is consisting of 146 base pair. These are very important points. DNA which is actually wrapping around the histone octamer, that is consisting of 146 base pair of nucleotides. So this is the total structure of nucleosome. This structure was considered as nucleosome. That means this is one nucleosome molecule. Now in this way this DNA will once again further be there, and this will once again wrap around some of the DNA, like this will wrap around some of the histones like this, and there will be the histones like this. Okay, there will be the histone octamer, this red colored portion, and once again this is the another nucleosome, and here once again the DNA which is wrapping around it, that is consisting of 146 base pairs. Now one confusion may come in your mind that in book you can find that there are 200 base pair in the nucleosome DNA, but actually that is this is the DNA which is also known as core DNA. This DNA is also known as core DNA, which is actually wrapping around the histone protein. And who will then what will be the where the H1 histone will go? So actually the H1 histone, the structure of H1 histone is like this. So this H1 histone will be like this, and this will actually hold the DNA thread around the histone octamer like a clamp. So H1 histone will act like a clamp which will actually join the DNA, hold the DNA thread over the histone octamer. So if this is the histone octamer, over it if I'll wrap one thread, then that will be this is the histone octamer, so there will be something to attach to hold that thread onto that position, that is the H1 histone. Now this DNA, this total histone, this is known as core histone. Let me just tell you few extra information. This histone is known as core histone. This histone octamer is known as core histone. This H1 is not coming into core histone. These, all of these, like this, H2A, H2B, H3 and H4, all of these are forming core histone. And the portion of DNA which is wrapping around the histone octamer or core histone, that is actually known as core DNA. So what about this much portion of DNA? There is also another H1 histone, and what about this much portion of DNA which is present in between two nucleosomes? This is one nucleosome, this is one nucleosome, and in this way so many nucleosomes are there. So what about this much DNA? This is known as linker DNA, which is almost consisting of 60 base pair, or different types of variation can come in the linker DNA only. This 146 base pair will remain always constant. There will be no change in the core DNA, that is 146 base pair, irrespective of any kind of eukaryotic cell. If you will see, then the core DNA will be consisting of 146 base pair. Only the linker DNA can vary. In case of human being, it can be like almost 60 base pairs. So 146 and 60 base pairs, or it can be less than 60, it can be more than 60, but only the variation will come in case of linker DNA. But in the nucleosome, there will be always 146 base pair in this much amount of DNA. And have you noticed that how this DNA is actually wrapping around the histone? This is actually wrapping around 1/3/4, like this kind of a position. This is the 3/4 part. This is this 3/4. This one and this one is the 3/4 part. And this is a one complete round. This is one complete round around the histone protein or histone octamer. So this is about the nucleosome model. Now let me just talk to you about this nucleosome model. When they will remain as it is, they will form the chromatin fiber. So now let me just tell you about the chromatin fiber, that chromatin fiber, and few more, these confusions are often arising in the mind of students. That is, what about the chromosome, what about the chromatid? So when these will be like this, once again just let me draw here. This will be like this, and the DNA will just wrap around there. DNA will just wrap around there. So these will form the chromatin. So in most of the time in the cell, we can find the chromosome or the DNA in this form, in the form of chromatin. When these will be further condensed, when the cell division will occur, when the time of cell division will come, then what will happen during that time? It is never possible for this much long thread to be divided equally, to be distributed into two daughter cells. Because what is the concept of mitotic cell division? That is nothing but the formation of same kind of or identical type of daughter cells like the parents. So the equal amount of chromosome must be there. So by that time the chromatin will further condense like this. So the chromatin will further condense like this, and sorry, let me just draw out here once again. They will further condense like this, and these will be the histone proteins around which they will be wrapping, the DNA will be wrapping. Okay, and in this way they will be further condensed to give rise to the chromosome. By that time the structure which they will give, that is known as chromosome, which we generally draw like this. We generally draw the structure of the chromosome like this. Then what about the chromatid? Now we all know that prior to cell division, during S phase of the cell cycle or synthetic phase of the cell cycle, the DNA replicate itself. That means DNA make its own replica so that the equal division is possible during cell division. So by that way, it will not be fitted within this kind of one single structure. So by that time they will make two copies of the same chromosome. There will be number of chromosome will be 1, but copy of this will be 2. So that when the cell division will occur during anaphase phase, these two chromatids will be separated and they will get detached from each other. They will go to the opposite poles. So these are nothing but the chromatids. These are the chromatids. So chromosome, chromatid, these are the structures, these are the chromatids, and or often we can say them as sister chromatid, and as they are attached to the same centromere, there is no individuality or there is no different situation. So we can say them as the sister chromatid. And when there is another chromosome, say for example this is another chromosome, and these are the chromatids of the other chromosome, then this portion and this portion, this chromatid and this chromatid will be considered as non-sister chromatid. So this is all about the chromatin, chromosome and chromatid. Now this structure, like when they will be further coiled and condensed, this nucleosome will be further condensed and coiled, they will form the supercoiled version. In that supercoiled version, almost 6 to 7 nucleosomes lie just beside each other like this. There will be the presence of 6 to 7 nucleosomes just beside each other, so that they will be much more condensed, that is very much condensed form, and after that they will be further condensed to give rise to a model which is known as solenoid. So this is the most compact form when there will be 1400 nanometer, or we can say that 1400 base pairs will be there in one round. In that way we can say. So this is about the solenoid, when these will be further condensed and very compact form will be formed. Then it will give rise to 1400 nanometer diameter of the chromosome, and that can be stained very easily and observed under microscope very easily. So this was all about the histone, octamer, histone protein and DNA and everything. So chromatin, basically it is made up of what? DNA and histone, which the scientist said, Roger Kornberg said, that this is the nucleosome. Around the histone the DNA is wrapping, and in that way The core DNA, which is consisting of 146 base pair, that will be constant for each and every eukaryotic cell. But the DNA which is present in between two nucleosomes, that is considered as linker DNA, that can vary from 60 base pair to less than that or more than that. So depending upon that variation, actually the main variation comes. And when the nucleosomes, that means these beads on a string structure, will get further condensed in so many much more way, that will give rise to 1400 nanometer width or length, that thread, that is actually known as chromosome, and that model is known as solenoid model. So this was all about the nucleosome concept. I hope this video was helpful to you. If so, then please don't forget to like, share and subscribe to my channel, and comment down below if you have any question. Thank you.